Hi, my name is Cheng En Xi, and I'm a student at McMaster University. Today I will be presenting a review of the animal and human trials of the AD5-nCoV vaccine candidate. On March 2020, the World Health Organization declared the COVID-19 outbreak to be a global pandemic. Since then, it has spread to every country in the world, resulting in more than 100 million confirmed cases and more than 2.4 million deaths worldwide. Vaccine development began shortly after the genome of SARS-CoV-2, the virus causing COVID-19, was sequenced. One such vaccine in development is AD5-nCoV by CanSino Biologics. It is a replication-defective type 5 human adenovirus vector encoding the gene for the SARS-CoV-2 spike protein. Adenovirus vectors have been previously used to develop vaccines for SARS and MERS, two zoonotic coronaviruses similar to SARS-CoV-2. Type 5 adenovirus, the one used in AD5-nCoV, is among the most common types of adenovirus. Replication-defective AD5 vectors have also been previously used in a tuberculosis vaccine trial, with promising results. Adenovirus vector vaccines contain DNA encoding the gene for the SARS-CoV-2 spike protein, enveloped in a modified adenovirus. The adenovirus will then inject the DNA into human cells, where the SARS-CoV-2 spike proteins will be produced. This approach is theoretically safer as it do not carry any real pieces of SARS-CoV-2. Adenovirus vaccines are also more stable than mRNA vaccines, allowing them to be stored at a much higher temperature. Antibody response to the receptor binding domain, or RBD, neutralizing antibody response, and T-cell response are used to assess the immunogenicity of the vaccine. Neutralizing antibodies are important as they bind to a virus in a way that prevents infection, as well as persisting in the body to protect against future infections. T cells are a type of leukocyte that falls into one of the two categories, CD8 and CD4, with the former being cytotoxic and the latter activating B cells for antibody production. The spike protein of SARS-CoV-2, responsible for mediating viral entry into host cells, is a key target for antibodies. The RBD of the spike protein has been found to play key roles in viral entry, thus it has become an important target in vaccine development. Key search terms such as AD5-nCoV, recombinant adenovirus, SARS-CoV-2, vaccine, human were used in varying combinations in PubMed to find all published clinical trial reports of the vaccine candidate. In addition, search terms such as animal, AD5-nCoV, vaccine, SARS-CoV-2 were also used to find all published animal trial reports. After the two searches, three published trial reports involving the animal, Phase 1 and Phase 2 trials of AD5-nCoV were found. Each report was then examined with a focus on the methods, results, and discussion sections for this review. All numerical data included in this review have a p-value of less than 0.05, making the numerical findings significant. The animal trial of AD5-nCoV was published by Wu et al. In Nature Communications on August 14, 2020. It was conducted on both mice and ferret models to determine the efficacy of the vaccine candidate on animals. Both mice and ferrets were also given high doses of SARS-CoV-2 virus following the administering of AD5-nCoV or a placebo. It was found that vaccinated mice had an increased immune response compared to those who were not. In addition, they also had no detectable virus in the lungs 5 days after inoculation. Among the ferret subjects, all vaccinated subjects produced spike protein-specific IgG antibodies and neutralizing antibodies at week 4, with no response in the control subjects. No virus was detected in the nose wash samples of the vaccinated ferrets 8 days after inoculation, while all control ferrets had infected samples. The data of this study demonstrated that a single dose of the AD5-nCoV vaccine candidate can completely protect the upper respiratory tract of mice and ferrets, as well as producing a good immune response. The Phase 1 trial of AD5-nCoV was published by Zhu et al. In The Lancet on June 13, 2020. It was a dose-escalation, single-center, non-randomized Phase 1 trial, aiming to assess the safety, tolerability, and immunogenicity of this vaccine candidate. A total of 108 adults with no SARS-CoV-2 infection history were selected. The participants were equally allocated to 3 dose groups: low, middle, and high. At least one adverse event was reported by at least 75% of all participants, with most adverse reactions being mild or moderate, and none lasting more than 48 hours. More than 90% of the participants in all dose groups saw at least a 4-fold increase in anti-RBD antibodies. At day 28, 50% of the participants in the low and middle dose groups and 75% in the high dose group had at least a 4-fold increase in neutralizing antibodies. This trial found the vaccine candidate to be safe and tolerable, with the low and middle doses selected to be further tested in the phase 2 clinical trial. The vaccine candidate was also able to induce immune responses rapidly in most participants. The phase 2 clinical trial was conducted by the same researchers, and its results were published in The Lancet on August 15, 2020. This study was a placebo-controlled randomized double-blind trial involving 508 adult participants. It aims to determine an appropriate dosage of this vaccine candidate to be used for a phase 3 efficacy study. It excludes those who have previous SARS-CoV-2 infections, major chronic illnesses, and pregnant women. 72% and 74% of the participants in the middle and low dose groups respectively reported adverse events, with 95% of all events being mild or moderate. RBD-specific antibodies were detected in both dose groups after 2 weeks. At day 28, seroconversion of neutralizing antibodies occurred in 59% and 47% of the middle and low dose groups respectively. Significant spike protein-specific T-cell responses also occurred in more than 85% of the participants in both dose groups. Following the results of this phase 2 trial, the researchers selected the low dose used in phase 1 and 2 trials to be used in a future phase 3 efficacy trial. Through both animal and human clinical trials, 85-NCoV has shown that it is capable of producing a strong immune response in both animal and human subjects, as well as being safe and tolerable to humans. It is capable of inducing RBD-specific antibody response and T-cell response, while the neutralizing antibody response being less adequate. The data published in the phase 2 trial supports the continued assessment of 85-NCoV at a dose of 5 times 10 to the power of 10 viral particles in phase 3 clinical trial. These results show that 85-NCoV is a viable vaccine candidate to be used against COVID-19, and its efficacy should be determined in a large-scale phase 3 trial. Here are my references, and thank you for listening.